round. And now we're going to the oral presentations. The first one is Helena Santos.
Hello, good morning. Let me share my presentation. Okay, so everyone's seeing my presentation, correct?
Yes, we can see it.
Yes. Okay, so my name is Helena Santos. I am a PhD student from the Universities of Lisbon in Portugal and Stellenbosch in South Africa. And today I'm going to present part of my PhD research entitled Putative aroma biomarkers of infected Vitis vinifera cultivar Trincadera with Botrytis cinerea. So, Trincadera is a Portuguese cultivar. It's a red grape cultivar that, as you can see in the photos, it's a compact cluster, average size and dark blue color. But it is highly susceptible to a disease called the gray mold disease provoked by the Botrytis cinerea fungus. And apart from that, it's also very irregular throughout the different vintage in the cultivar. So it makes very not constant wines, but with a lot of potential for being quite tasty. It's cultivated mainly in Alentejo or in the north in Douro and Down region. So this cultivar has the potential for good wines, but it's not considered an aromatic cultivar. What does that mean? So as we saw a bit in Dr. Francesco's presentation, volatiles are part of the secondary metabolites of plants, and they contribute with different aromas to wines and grapes. And different cultivars will have different types of volatiles. In this case, Trincadera doesn't have a lot of them. So for example, we will not find terpenes family in Trincadera, but we'll find others like, for example, the green leaf volatiles and some esters. It's also relevant that in the volatile metabolism and balance, some volatiles will be found in their free form, which means they are ready to be used by the plant to be released, or they can be found in their glycosylated form. And therefore, they will be complexed in sugars or amino acids so that they are not being used by the plant. This happens because they might have some toxicity and are involved in pathways that are not necessary at the moment of the plant. So we wanted to see how the volatiles of Trincadera, both free and glycosylated, change upon an infection by the Botrytis cinerea. For this analysis, we had two approaches. The first was the volatile identification. So we harvested 4 clusters, healthy clusters, and 7 symptomatic infected clusters by botrytis. We made juice out of this, and part of the juice was run in GCMS for the free volatile, and the other part of the same cluster of the juice was treated with beta-glycosidase to release and cut the sugars that complex the glycosylated volatiles. And therefore, once they were free, they were again run for the GCMS identification and GC feed for quantification. A second part of our work was as well to freeze and make a thin powder of this grape so we could extract RNA, synthesize cDNA, and then through qPCR, analyze the expressions of the genes that could be related in metabolism of specific volatiles. So first results, I show you a multivariance analysis, an OPLS, where we can see that control samples were clearly separated from infected samples. So this shows that there's already a difference in the volatiles between one and the other. And the drivers for this separation in the case of the control samples are mainly volatiles from the ester fatty acid family. And in the case of the infected samples, it were ester acetates. As the principal component analysis showed promising results, we went in a further statistical analysis for each volatile separately. So regarding the free volatile fraction, What we can see is in this plot that in this blue box, we have two compounds that are statistically changing in infection. These two compounds belong to the LOX pathway, so the green leaf aroma volatiles. And as well, Dr. Francesco said, they are known for their role in defense. And they are also associated in a pathway for gismonic acid, which that we can see here already that infection is triggering a defense mechanism in the grapes. We can also see here in the red boxes that 2-phenylethyl acetate and exilic acetate are not present in control samples, but upon infection, they appear and we can detect and quantify them. Like I said, Trincadera is not an aromatic cultivar, so we don't see many free volatiles. But if we look at the glycosylated volatiles, the story is now different. So all of these were complexed in sugars and were released with the treatment. What we can see, first here, alcohols, amyl alcohol, 2-phenylethanol. Alcohols are known for their disinfectant properties. The fact that they are increasing in infection show an attempt of the plant to defend themselves against botrytis. We can also see here, which is our most interesting result, I would say, neither of them are present in control samples, but in infection they spike them. So we have a total new family appear right of the plot. Control. Their relative precursors, so for example, ethyl decanoate acid, so their precursors are increasing, which are fatty acids. Fatty acids are still being studied, but there's the chance or like the hypothesis that they have a putative role in inhibiting Conidia germination of Botrytis cinerea. On the other hand, it is known, or it is being studied, let's always say there's putative chance that ester acetates potentiate Conidia adhesion of Botrytis cinerea. So you can see sort of a balance of a plant trying to fight botrytis and botrytis trying to modulate the plant for its own development. Regarding the genes expression, we tested 21 genes and only one gene showed a difference in the expression, which was the HPL2, which is a gene involved in the LOX pathway. It codes for an enzyme that will lead to the volatiles that we saw there were change in the free fraction. So there's connection and we can correlate the metabolomic and the genes build up this model of putative behavior of the grape Botrytis cinerea. So in sum, upon infection, and we can see that then we have the ester acetates increasing only in infection. We have the acids, fatty acids only increase detected in infection, as well as alcohols that they are all increasing, and these two were not in the plot due to scale reasons because they are very high, as well as heptanol, as you can imagine. It's one of the most common volatiles, so they were just out for that reason. But as we can see, we have many compounds that we can call them positive or negative markers of infection, at least in Trincadera, infected by Botrytis cinerea at the time point there was the harvest grapes. It's also interesting to point that the ester fatty acid exilic xanoate completely disappeared in infected samples. So that shows that their decrease can lead also to disappearing. So this could be a negative marker of infection if we can verify this in different experiments, new vintage, etc. So the majority of them in infected varies. And infection is indeed leading to a LOX pathway activation, whether we don't know if it's to promote as well gismonic acid defense or it's for the green leaf volatiles signaling and communication with plants, etc. But it's the possibilities. Once again, we didn't see terpene, so that corroborates that Trincadera is not an aromatic cultivar. The majority of the volatiles were in their glycosylated form, so without the treatment with beta-glycosidase, the study would have been much simpler and less informative. And that we have these changes that we don't know if they are a botrytis manipulation for development or a plant cell defense mechanism. And this balance, it's unknown, but it's very interesting to understand, and it would be even more interesting to see what happens in other cultivars or in different vintages. Like I say, is this behavior, are these markers of infection specific, and can they be used in the industry? So these are the questions that we end our work with that, well, we need to further explore in the future. But let's just say that Asterostate family are very promising in Trincadera for infection in Botrytis cinerea. Well, I would like to thank everyone from my group that work with me, and thank you for accepting my presentation. I'm very happy to be here and ask any questions you would like. Thank you.
Thank you, Helena, for your presentation. The questions will be after everyone. So, but if you have any questions, please write it for you to don't forget to make them. So next up is